Okay, so in most classes, some left off right here with this picture. So in some classes I talked about this picture, some classes I didn't. So I'm going to start with this. If you were a class that we've already talked about this, you can just fast forward to where we left off. So this just gives you, if you remember, we were just talking about how a virus infects a cell. And so here's the virus, and this is the capsid around it, and then you can't see it here. There's your DNA inside in blue. And so then what happens is the capsid itself is colored here, all right, breaks up, all right. So this is the capsid is broken down. All right, so that's this arrow here is the broken down capsid. And then this arrow here is the other part of the virus, which is the DNA. So DNA is double-stranded, so there's your viral DNA. So now there are two pathways, or two things that I should say that the DNA is used for. So thing number one is to replicate the DNA. All right, so we get lots of viral DNA. And so we could say here that that's going on with replication here. So the host cell enzymes replicate viral DNA. Okay, so that's what's going on here. So viruses don't have their own enzymes to do that. The second thing is going through this process here. So notice here, DNA to, let's get a different color, mRNA. That process, DNA to mRNA, is transcription. Collectively, this whole process is called protein synthesis. From right here to right here, we're using the DNA from the virus to make viral proteins. And so I'm going to keep along with that color. So these guys here, all these are pink, like the capsid protein. So they're used for the capsid protein. And this right here, this would be transcription going from DNA to mRNA, and from mRNA to protein, that would be translation. Okay? And so then what happens is all the DNA and the capsid proteins come together to make your new viruses. So your new viruses have the genetic material, and they have the capsid, and these guys now can go infect other cells. So they can go infect other cells. So the virus uses one cell. That cell, viruses can do a couple of different things when they infect a cell to get new viruses. Some viruses kill the cell and produce hundreds of new viruses that go and infect other cells. Some viruses incorporate their DNA into the host cell's DNA and can kind of stay put there, and therefore always kind of be able to make viruses. So we're going to look at some different types of viruses and how they work differently. So the first one is the bacteriophage, viruses that infect bacteria. So these guys are unique in that they go through a couple of different cycles. They use what's called the lytic or the lysogenic cycle, or sometimes both. So let's talk about the lytic cycle first. Results in the death of the host. In the last stage of the cycle, the infected cell lyses, which means, remember, to break open. So it's going to kill the host cell and releases the viruses produced within the cell. Each virus can then spread to infect other healthy cells. So it's like this picture, except it gets so chock full of viruses that the host cell explodes, and then all the little new viruses come out and infect cells. So I want to show you a little animation of what happens. So here's a bacteriophage coming to infect a bacteria cell. And what it does is the little tail fibers bind to receptors on the cell that it's infecting, and and that's what allows it to infect that particular bacteria. Then it's going to eventually inject its DNA. So let's look at what happens here. So there it is. If DNA is the genetic material, infecting the DNA. Now look at what happens to the big chromosome of the normal cell. It destroys that larger chromosome that belonged to the cell itself and then uses the bacteria to make many copies of bacterial DNA. Now then what is DNA used for? DNA is used to make proteins, right? So then what happens is that DNA is going to make and allow the bacteria cell to now make viral proteins and replicate the DNA. And so therefore now we have new viruses, the cell lyses, and guess what? Those new bacteriophages can go and infect new cells and so on. So it sacrifices one host cell, but you get hundreds of bacteria possibly in the process, and then that can go infect a different cell. And so therefore we have this cycle. The top left is the first one part of the cycle, and you go around the cell lyses, and then those new bacteriophages can go and infect new cells. So that's what you see here. So this is the lytic cycle. So that's the first part of it. It'll inject its DNA, right, into the host cell. So or it's genetic material, I should say, because it doesn't always have to be a DNA. And it breaks up, as it says, degradation of the host DNA, makes new host proteins, viral proteins, puts them together, lyses, and can go and assemble a new bacteriophage. Okay, so that is the lytic cycle. So now let's take a look at the lysogenic cycle. This, the viral genome replicates, but does not, destroy the host cell. So an example of this is the lambda phage. That's a particular type of virus that infects E. coli, and it does both types of cycles. It goes the lytic and the lysogenic. So infection of an E. coli by lambda begins as usual. What's as usual? Injecting into its genetic material. And then what happens next depends on the reproductive mode that it's in. So if it's going to do a lysogenic cycle, so during a lysogenic cycle, the viral DNA molecule is incorporated. This is what I was talking about. It can become part of the host DNA, incorporated into the host cell's genome. Then when the host divides, it copies the phage, or the viral DNA, and passes the copies to all the daughter cells. So what happens is the viruses propagate or replicate or make many more viruses without killing the host cells. All right, so what happens is, if this is your normal bacteria and this is the normal chromosome, when the virus injects its DNA into it, so the DNA goes in. What happens here, instead of what we just talked about in the lytic cycle, where it uses and starts making viral proteins and so on right away, what happens is in that big chromosome, remember bacteria have one big circular chromosome, you have the viral DNA. So then every time this virus, or this bacteria, reproduces, you get each new bacteria now has viral DNA. And guess what? That viral DNA at any point can pop out and be used to make new viruses and actually go through the lytic cycle. So this is where it says the viruses may enter the lytic cycle at a later time, usually triggered by an environmental stimulus. It could be, an example, chemicals in the environment that might trigger it, a different pH, something like that. And so I want to show you another video or a little animation about the lysogenic and lytic cycles and how they relate. Okay, so let me hit replay here. All right, so unlike the lytic cycle, the lysogenic cycle reproduces the viral genetic material without destroying the host cell. So here comes a lambda phage, and it binds to the surface of the bacteria, injects its DNA into the host cell, leaving the empty protein coat. You saw that didn't go in, and the viral DNA is actually incorporated into the host cell's DNA, and it's called a prophage when it does that. And so then when we look next, every time the host bacterium now reproduces, it replicates that phage DNA along with its own DNA, and so therefore you can have very quickly millions of bacteria that have the prophage in it and can anytime do the following. So occasionally the phage DNA exits the bacterial chromosome and enters what's called the lytic cycle that we talked about before. So now the viral DNA takes over and is used, it's replicated, makes proteins, and the cell lyses, and now all those little bacteriophages can go and infect new cells, and then those new cells can enter the lysogenic cycle. So the lytic and lysogenic cycle, some bacteriophages can go back and forth between the two. All right, so let's look back at our notes. So if we look at here, that's what this picture is showing you here. So this is the lytic cycle that we talked about before, where it injects the DNA. All right, so you have the DNA, it cuts up the bacterial DNA, so you don't see it in here. The bacteria uses the DNA, viral DNA, to replicate and make more proteins, and lysis occurs. Now over here is what happens. We still have the same starting point, so this is where we start, and everything goes through here where the genetic material is entered in, and here it is entered in. Now it's what happens from there. So this is the fork in the road here. All right, so the lytic cycle is induced, or the lysogenic cycle is entered. So if it goes into the lysogenic cycle, it incorporates that viral DNA into the bacterial DNA, and that's your prophage. And so then what happens is then every time the bacteria divides, it replicates the prophage DNA, and therefore has it in every single bacteria cell. So you can have millions and millions and millions of bacterial cells that have this. And then occasionally, if the environment is right, anything can trigger that prophage to pop out of the bacteria cell, and then it can enter back into the lytic cycle and therefore kill the host cell and make new viruses that actually can go and infect more cells and do the same thing over and over and over again. So bacteria, like us, even though they're only one cell big, they have some defenses against viral infections. And so let's talk about a few of those. So phages can wipe out an entire bacterial colony. All right, remember, a colony is a group. It can be of millions of bacteria. Bacteria are so small that you can't see them, but when they divide and you get millions of them, they look like a little circle, kind of, but there's millions of smaller organisms within there. So natural selection favors bacterial mutants with the receptor sites that are not recognized by phages. All right, so if you have a bacteria, and this would be a good mutation. A mutant is an organism that a mutation has occurred, and bacteria only have to have one mutation and to change the whole organism because they're only one cell big. So if that mutation actually changes the receptor sites on their cell membrane and the phage doesn't recognize it, then all of a sudden that particular bacteria can't be infected by that virus, and therefore that's a good thing, and that bacteria is going to live and divide and make more bacteria like it, and then all of a sudden you have a new phenotype within the population. So we see mutations affect bacterial colonies and bacterial populations much quicker than in multicellular organisms like us, because the mutation to go to the next generation, us, has to be in an egg or sperm cell. So bacteria produce what are called restriction endonucleases. ACE, it's an enzyme, otherwise known as restriction enzymes. They naturally have these, and what it does is recognizes and cuts up foreign DNA, including phage DNA. So if the restriction enzymes get to it first, when that bacteria or that virus enters their DNA into the bacteria, restriction enzymes may get to it and break it up before it actually becomes part of their chromosome, or it may not. So that's one of the defenses. And this is important. We'll see in chapter 20 that we utilize our knowledge about restriction enzymes to manipulate DNA. So natural selection favors phage mutants that are resistant to restriction enzymes. So if you have a virus mutant, the mutation has occurred, that happens to not be, their DNA not be cut up by restriction enzymes, that's a good thing, all right? And so therefore, that's a good thing for the virus, bad thing for the bacteria, okay? And so, so there are some defenses, even though they don't have a souped up immune system like we do, because they're only one cell big. So let's look at another type of virus. So we've looked at the bacteriophages, only infect bacteria. So let's look at some of the viruses that infect eukaryotic cells. So envelope viruses, and I introduced this with the influenza viruses. Viruses with an outer envelope use it to enter the host cell. So it's a way to get inside the host cell. What happens is the envelope fuses with the host membrane, moving the capsid and viral genome inside. So let's turn it over here. After the virus assembles, it buds from the host cell. So let's look at this. If we look at down here, this particular picture. So what you're seeing is an envelope virus like the influenza virus. So if we go with the same color coding, here's your capsid. And so the capsid is pink. And then this is an RNA virus, so the genetic material is RNA. So that's your RNA. And then you have your envelope around it, all right? And the envelope has glycoproteins sticking out of it. That's these guys here. That's going to help to bind with the surface of the cell, all right, of the host cell. So let's draw that here. So this comes over here, and I'm just going to just show you this like this. It's going to go in like endocytosis, where it's going to fuse with the membrane and allow its insides to come into the cell. So this is the insides, is the capsid and the RNA. All right, what it's not showing you is, like we looked at the generic picture over here, is remember the capsid breaks up and then you end up with your genetic material exposed. And so that's what we have here is the, draw here, capsid proteins broken up, capsid broken down. And then you have, here's your viral genome. All right, so this is an RNA virus, okay, with RNA as the genetic material. And so what happens next is this RNA is used as a template. What do we use a template for? We use it to, like if we think about the DNA and what side of the DNA is used as a template, it's this template that you are building something off of. So we're going to make new mRNA. And that mRNA, let's get a different color here. I'll use red. All right, this shows you here an arrow showing you that it's being made. So let me give you an example over to the right about what's actually happening. So let's say you have your viral RNA is AUGCCU. This is your viral RNA. So that's this right here. And so then what's happening is we are kind of quote unquote making a copy. That copy is going to have complementary bases. So it would look like this, U, A, C, G, G, A, and so on. So this is the mRNA that now is going to be used. Now notice a couple things going on here. All right. pathway 1, the mRNA is used for protein synthesis. So, for protein synthesis. That's this part here. Let's label this 1, used for protein synthesis. And so it's going to make proteins. So let's look at let's use the pink with the viral proteins again. So it makes new capsid proteins. All right, so that's these guys here, using that mRNA. It also makes glycoproteins, because remember, the glycoproteins here are part of the envelope. And so so one of these sticking out is a glycoprotein. So this is made on ribosomes in the ER, and what happens is you get these little glycoproteins in the inner part of the ER. But if you remember back when we talked about the endomembrane system, that from the ER you get a little vesicle that buds off, like this, that buds off of this. And it has these glycoproteins inside, but look at how it gets outside as it fuses with the membrane, kind of like exocytosis. And now the membrane on the outside of the host cell has these glycoproteins on it. All right, that's going to become important in just a minute. So we're making the capsid and the glycoproteins. That's thing number 1 that we use the mRNA for. The second pathway, this way, was to make more copies of the RNA. So remember, we use the red, so we're using the red as here, which is this one. So the red was UACGGA, so that's your mRNA. And we're going to make new RNA that's going to, we want the viral RNA, and so that's this here. So what is that going to look like? AUGCCU to go with this. These two go together, they're complementary. And this is the RNA. See how that when they used that template that was made, it had the opposite bases. So now that when you make the mRNA again to replicate it, to put into all the new viruses, it's the same order of bases as the original RNA that came into the cell. And so therefore that's your copy of the RNA, and now all your capsid proteins come together and circle the RNA. And then how does that capsid get out? By exocytosis. But look at when it buds out, the cell membrane is around it, and what does the cell membrane have as a result of the ER? It has the membrane around it and all those little glycoproteins. And so now you get a new virus that is exactly like the first virus. All right, so you have the capsid, and you have your DNA inside, and you have all your glycoproteins sticking out, and that new virus now can go and infect a new cell. All right, and so therefore, and that's how an envelope virus works. So that's why when we wrote the envelope viruses, the envelope helps them to get into the cell. This is how, because it fuses with the cell membrane and then uses the host cell machinery, in a sense, to make viral proteins and RNA. So that leads us right into RNA viruses, where RNA is the genetic material like this particular picture. We're going to look at a specific type of RNA virus called a retrovirus. So retroviruses have the most complicated life cycles. So this is a type of RNA virus. This is not what I just did here. So this is a typical RNA virus. We're going to look at a special type. These, why they're special, they have another enzyme called reverse transcriptase. Let's look at that word, reverse transcriptase. Transcript, coming from transcription. ACE is an enzyme. Reverse means the opposite of transcription. So what is transcription? Transcription is taking DNA and making RNA. And so then what we're going to do now is take RNA and make DNA, which is the exact opposite of transcription, hence the name reverse transcriptase. So that transcribes DNA from an RNA template. So we're going from RNA to DNA. And HIV, big retrovirus, all right? So HIV, human immunodeficiency, HIV virus. All right, this is the virus that causes AIDS. AIDS is the disease, so the acquired immunodeficiency syndrome. And this virus that causes that is what we call a retrovirus. The virus itself attacks certain cells of the immune system. It can weaken the immune system, so it's difficult to fight off infections. And so normal infections with a healthy immune system are not life-threatening. If you have HIV virus in you and it begins to weaken your immune system, and therefore you are more susceptible to diseases or infections that people who aren't infected with HIV and have a healthy immune system can normally fight off. So people usually die of something else besides AIDS, like they'll die of pneumonia, but it's because they're not able to fight off the infection when they should have been able to, because they're a compromised immune system because of the HIV virus infecting it. So they can be life-threatening in someone with AIDS, which, as I said, is the disease that results from HIV infection. So this shows you the HIV virus infecting a cell. This is a membrane virus, so it invades the cell much like the previous picture that we looked at. Let me just talk a minute about the HIV virus. And so it has a membrane envelope, and on the envelope it has sticking out, like the previous picture, you have these glycoproteins, and I'm not going to draw it all the way around. You can see it all the way around here. So these are glycoproteins. These guys help them to get in, to bind to the surface receptors. And this is the envelope, or the membranous envelope, that's going to help them to bind in there. Then, if we look inside of here, we have this part right here that kind of looks, this part right here. It's this part right here. This picture is hard to see, so I just want to make it bigger so that you can see. Actually, I should probably color code this part too, showing it how it's getting into the cell using the envelope. And then you have your two pieces of RNA. So HIV is an RNA virus, so you have two pieces of RNA. So I'm going to put here that these guys are two identical strands of RNA. So RNA is the genetic material. And then what's unique is that it has this enzyme. What was the enzyme called? Reverse transcriptase. That's the blue here. This is the reverse transcriptase. Okay, so if we color code this part here, you have your two RNAs, and then you have your reverse transcriptase. It's kind of hard to see there. Okay, so what comes in? What we see here, all the little dots are the capsids. So we see the capsid proteins being broken down. All right, so that's similar to what we've talked about. And we see the two pieces of RNA. So that's this right here, two pieces of RNA. And attached to it that you can't really see is the reverse transcriptase. So you have the two reverse transcriptase enzymes. So now that's all in there. So now you have RNA. So let me just give you the big picture. What a retrovirus does, we as a host cell, this is the cells of our immune system, our genetic material is DNA. The goal of the HIV virus is to insert its genetic material into our genetic material, into one of our chromosomes. And so our chromosomes are made out of DNA. RNA is the genetic material for this virus. So therefore, this is why the reverse transcriptase is so important for the virus, is that the reverse transcriptase, once in a host cell, can actually be used to kind of copy the genetic material from the RNA into a molecule of DNA and then insert it into the host cell's chromosome. So that's what we're going to look at here. So I was using red for the viral RNA here. So here's one strand of the viral RNA, and here is the reverse transcriptase, which is going to do the reverse transcription. I'll underline these with the color coding here so we can see. So that's what came in from the host cell. So what it actually does is actually make DNA from that. So here's your original RNA. And so let's say the RNA is AUGCGG. So this is the RNA from the virus. So what we're going to do, or what the cell does, is, let's do a different color here, let's do purple, is use that to put the matching bases, but not RNA bases, DNA bases, or nucleotides here. So we're going to build a DNA strand from that. So the DNA that would match up with the order of bases in the RNA, DNA, what goes with the A? You'd have a T, since it's DNA. What goes with the U? A, C, G, C, C. So that's your DNA. So this is your DNA in purple, RNA hybrid. So we use the RNA as a template to make the DNA strand. If you know one side of the DNA, can you figure out the other side of the DNA? Absolutely. And so what we do is we take that single side, the TACGCC DNA, and we make the complementary side. So ATGCGG. And now we have a double-stranded DNA with the coding pattern that was in the RNA of the virus. And now that DNA, that corresponds to it's the same genetic material, DNA, that we use. So now look at what happens next is that DNA gets incorporated and goes into the nucleus, goes within the nucleus, and becomes part of our DNA. So I used purple here, so we're going to look here. So it's part of our DNA now in one of our chromosomes. And so so let's write that here. Let me write it along the side. Let me get a just black pen here. And it's called a provirus. All right, so let's write here that what's going on is the DNA is incorporated as a provirus into the cell's DNA. I'm going to put a little star here that this stays within the cell. So this is part of this cell now. So if the cell divides and make more cells, it's going to have the HIV virus DNA in it. So that's a provirus. That's what that says there. It's hard to read. Okay. So then it stays in there, and then eventually, at some point, it can be triggered to now go through protein synthesis and replication and make new viruses. So that's what we see here is this is your DNA. What can we do? With DNA, when we go through transcription in our nucleus is make mRNA, right? So here's your RNA from your DNA. That's called transcription. Going on there. And so we're using not our gene, but the virus's gene, and we're making the RNA. Now notice two pathways here. The RNA can be used for, it says the RNA genome for the next viral generation, so that's thing one. Or the RNA can be used to make more viral proteins. All right, so what are the viral proteins? The capsids. So here's the capsids. So it makes capsid proteins and the glycoproteins, like we talked about in the ER earlier. So this is the ER. And there are ribosomes on the ER that then are used to make glycoproteins. So mRNA to proteins, that process, this right here, this process right here is translation, right? Making proteins. This is transcription. Some of that RNA never is used to make proteins, but is used as the genetic material. Some are used to make the viral proteins. And remember, the vesicles butt off the ER. That's what this is showing you here. Vesicle butting off the ER, fusing with the membrane, and now the outside of the host cell, our cells of our immune system, have the capsid proteins, the glycoproteins, and now the capsids and the RNA come together. So here's your capsid in pink, your proteins right there. Here are your new RNAs. And I should say that they also use it to make more reverse transcriptase. And then that gets incorporated into a new virus. So we have this right here, two RNAs. Membrane with the capsid protein buds off via exocytosis, and now we have a new virus that can go and infect other cells. And the beauty for the virus is that this cell stays intact. It's not dead. It still has the viral DNA in it, and therefore can do this again and again and again and make new viruses, and therefore the cell is forever infected and can always make new viruses. So good for the virus, not so good for the host cell, which is the cells of our immune system. So that's how retroviruses work. And so let's go ahead and I'm going to put here that they also make reverse transcriptase. So these are capsid proteins, and I'm using the same color here. They also make new enzymes, reverse transcriptase. The enzyme gets put into each one of these, so now they can do the same thing to the next host cell. And so retroviruses in a little class all on their own, but based upon the way that they infect, and so in having that particular enzyme. So the next section is just some basic things about viruses and diseases. So I'm just going to go through it kind of quickly here. There's not a lot of fill in the blanks and things like that, but I just wanted to bring in some stuff from what we've talked about with the immune system and going with with vaccines and things like that. So some viruses damage or kill cells by triggering the release of hydrolytic enzymes. Comes from hydrolysis, right? Which means to break down. By adding water. And so from lysosomes. So viruses can damage or kill cells by doing that. Some cause the infected cell to actually produce toxins that lead to disease symptoms. So the toxins can, from the virus, can lead to that. Others have molecular components like envelope proteins that are toxic. So the proteins on the envelope is what makes you sick. In some cases, viral damage is easily repaired, like damage to our respiratory cells. The epithelium is the tissue on the outside of our respiratory system that gets infected. So we have a lot of symptoms because of damage done to those cells, but we can replace those damaged cells fairly quickly as we destroy and get rid of the viruses with our immune system working. But in others, infection causes permanent damage, like for instance, polio actually causes irreversible nerve cells to be damaged, and so therefore you have long-lasting effects as a result of that. Many temporary symptoms associated with the viral infection result from the body's own efforts at defending itself against the infection. And so, example, All right, many of the symptoms, example would be fever. So the fever is your body's way of heating you up to destroy whatever's infecting yourself. So the immune system is part of the body's natural defense against both viral and bacterial, lots of the different infections. So if you remember, vaccines we talked about are harmless forms or parts of pathogens that stimulate the immune system to mount defenses against the actual pathogen. So you remember your memory B cells and T cells, and those memory cells then stick around, and therefore the next time you come into contact with that pathogen, you can fight it very quickly and usually don't get sick. So some things here, vaccination has eradicated smallpox, which means that virtually used to be a big deal. A lot of people would get it, and now a lot of people don't. All right, effective vaccines are available against, and so we have a lot of these, polio, measles, there's mumps, rubella. That's the MMR vaccination that children get. Hepatitis B, HPV, and a number of other viral diseases have vaccines. Some viruses do not yet have effective vaccines, and so they are currently always working on vaccines and trying to make them better. So an example, the influenza pandemic, 1918 to 1919, killed more people than World War I. And so it's just an interesting fact that a pandemic of virus that we don't have a vaccine and can be very, very deadly. And so things that people used to die of or, be treated for, 100 years ago, people don't die of that anymore because of the vaccines. It was the most devastating epidemic, the influenza pandemic, in recorded world history. More total people and proportionately more people died of influenza in the single year than in the 4 years of the Black Death or bubonic plague that happened in the 1300s there. So it was a big deal, and so like I said, we don't see those pandemics anymore because people have really worked on getting vaccines for viruses and bacteria that are particularly harmful or seem to affect a lot of people in the population. So medicines can do little to cure viral diseases, and so we can just treat the symptoms, but there's not a lot that we can do. So when somebody gets a cold, you just have to write it out and your immune system works against it. Antibiotics are powerless against viruses. They only work on what? Bacteria. They destroy the bacterial cell wall. So they are ineffective against viruses. There are antiviral drugs, that try and blocks. They can't quote unquote kill the virus. They don't destroy the virus, but it works on stopping the virus from reproducing. So most of them interfere with the viral nucleic acid synthesis. So meaning nucleic acid, RNA, or DNA. So depending upon what kind of virus, if it blocks it or stops it somehow from replicating its genetic material, then it can't reproduce in the host cell. So this is blocking it in the host cell because they can't reproduce by themselves anyway. So an example is this drug that impedes the herpes virus reproduction by inhibiting the viral polymerase that synthesizes viral DNA. And so therefore, if it acts as an inhibitor to the polymerase, then you can't make more DNA, and therefore you don't have the genetic material for inside the viruses, and the viruses can't be reproduced inside the cell. AZT is a drug that's used for HIV reproduction, so it curbs or stops or slows HIV reproduction by interfering with DNA synthesis by reverse transcriptase. The same idea, going back to this picture, is it stops this part here. So if reverse transcriptase can't work, you can't make the DNA, which means it can't be inserted into the cell, which means that it can't make new viruses, and you really slow the infection down in the body because less and less host cells are going to be infected. And so currently, especially for a lot of these viruses, and especially HIV, they have these multi-drug cocktails where it's it's a mixture of different types of drugs that attack and stop, going back to this picture, different parts of how the HIV infects the cell, and so those collectively altogether seems to be the most effective treatment. So it's not one drug that they're taking, but a collection of different drugs. And this is where I would like to stop.